In this lecture, we will discuss about the interaction of proteins with other molecules. What are the functions of the proteins interacting with other biological molecules or small molecules, and how to identify the binding sites, and are there any specific preference of interactions at the binding sites. In the last class, so what did we discuss in the last class? So, we mainly focused on protein folding rates, right. What is protein folding rates? It is a measure, right, to see whether a protein can take less time or more time to fold from its amino acid sequence to 3-dimensional structure, right. It is order of 8 order of magnitude, right, from milliseconds to an hour, right. So, there are various parameters, right, which can relate the protein folding rates, specifically the interaction between amino acid residues in protein structures, for example, the medium and long range interactions, right. So, the medium range interactions influence, right, mainly in the case of all alpha proteins, and the long range interactions in the case of all beta proteins, and we see that the all alpha proteins fold faster than all beta proteins, right. Likewise, we related several structure based parameters, right, to understand the protein folding rates, right. So, what are various structure based parameters we discussed in the last class, right, the contact order, long range order, right, the multiple contact index, right. So, we derived various parameters and related the parameters with folding rates. We showed that these parameters, right, have inverse relationship with the protein folding rates. Then we try to utilize these parameters to predict the folding rates as well as predicting the folding rates from amino acid sequence in two different ways. One is directly we can use the properties, or we can predict the contacts, and from the contacts we can calculate the long range order and we can get these folding rates, fine. So, today we will discuss about the protein interactions, right. Proteins interact with several other molecules, right, biological molecules and the chemical compounds, right, for various functions like protein interactions, protein RNA interactions, right. So, protein DNA interactions, so here you can see the protein as well as this is a protein here and this is RNA here. So, this is a protein RNA interactions, right. You can see the protein here, right, and you can see the DNA. So, this protein DNA interactions, right. Here you can see the proteins, right, and see the ligands here. So, this is protein ligand interactions, right. So, these interactions are important, right, for various cellular processes, right. For example, if you take the protein interactions, so they are involved in various processes, for example, cell signaling, right. This is a complex system of communication, right, which governs the basic cellular activities as well as the coordinating cellular actions. Ubiquitination, so this is a post transcript modification, right. So, this also affects the activity and promote or prevent the formation of these protein interactions. So, also the several protein complexes, they are acting as molecular switches, right. These interact with the different proteins with a varying specificity, right. So, for example, same protein A interact with B with specific specificity as well as with C with a different specificity. In this case, they have the bias, right, to which molecule it can interact with high specificity. Likewise, antigen antibody interactions, right, there is most of this immunological process. So, you can see the interaction between the antigen as well as this antibody. So, for understanding the invading the bacteria and the viruses, fine. Likewise, in protein nucleic acid interactions, right, these protein nucleic acid interactions are important for the regulation of gene expression, DNA replication and repair, also transcription, packaging and so on, right. So, we look into these protein ligand interactions, right. These ligand interactions are also important because these ligands trigger the affinity of these compounds, right. This can interact with the active sites, right, and change the conformation of the protein, right, and then they inhibit several activities. So, the protein ligand interactions are important in structure based drug design, right, for identifying these lead compounds, right, or the potential inhibitors for various targets, right, which are involved in different biological pathways as well as in different diseases. So, now we have these different complexes, we have protein protein interactions, protein DNA interactions, protein RNA interactions and protein ligand interactions. How to extract the information, right, from known structures, right. So, these structures are deposited in protein data bank, right, and how to obtain the structures, right. For example, if you are interested in protein protein complexes, specifically on the heterodimers, right. What is heterodimers? Different. Yeah, the two chains which are different, the molecules are different, right, either can the dimer or trimer and so on. So, in this case we go to this protein data bank and the advanced search options and see this contains protein, right, yes or no. It's yes, because we are looking for the protein protein complexes. Then it contains DNA, no DNA, right. So, RNA, no RNA, then the hybrid, that is we do not need. Then it contains protein means you can get all these structures with proteins, right. Sometimes you can get the monomers and the dimers and the same proteins, right, as dimers, trimers and so on. So, now we have to define how many chains do you need, right, as how many asymmetric units, right, at least we need 2, right. Then you get to see the number of chains based on biological assembly, right. Here let us say we take 2 and then if you submit the query, right, then you will get the data. So, you can see this is the heteromer, this is the homomer, right, and you can see how many monomers and other choices. So, then we will give the list, right, and then you can get the structures, that you can see the structures and you can carry out your analysis, right. For example, if you have protein protein complexes, right, for example, here it is A is one protein and B is another protein. So, these proteins A and B, they form the complex, right, AB complex, and if you look at these structures you can see the some regions where these proteins can interact, right, for example, this region, right. So, you can this can interact and this region, right, we call as binding sites, right. So, then what is the binding site? Interact, right. So, there is a residue in a protein where, right, these two proteins are interacting, right, this is called the binding site, or you can say interface residues, right, because they are sharing the common interface between these two proteins. So, now the question is, okay, there are several complexes, protein protein complexes, protein DNA complexes, protein RNA complexes and so on. How to identify the binding sites, which residues are located at the interface area, right. So, there are various methods to identify the binding sites, right. The major method is you can use the distance based criteria and you can use the soluble accessibility based criteria and also you can use energy based criteria, right. So, how to identify the binding sites based on distance, right. For example, if you see this is the protein DNA complex, right, here I shown example as protein protein complex, right. How to identify the binding sites? If you look into these structures we can tell, right, because here you can see this is a region where the DNA can interact, right. Here also you can see here this is the area where the proteins can interact, but how we quantify, how to exactly identify which residues are interacting, which residues not interacting, right. For having the uniform representation of these binding site residues, right, you can define the binding sites using distance based criteria. For example, either you can use the C alpha atoms or you can use C beta atoms or you can use any heavy atoms, right. Based on that we define the distance, right, between these two atoms. For example, if you take the C alpha atoms, right, generally they use the distance of about 6 angstrom. In the case of C beta atoms, here also 6 angstrom is used, and any heavy atoms that depends upon the type of the complex, right, you can change the distance, right. So, for example, if you say 5 angstrom, right, let us see any heavy atoms, right, which is a distance of 5 angstrom. You can use different criteria. If you understand the binding site residues and the type of interactions, which one will be better, which one gives the best data, either to use C alpha or C beta or any heavy atoms, like any heavy atoms, because if you want to see the electrostatic interactions, that is between the atoms, right. So, the oxygen in the negative charge residues and the NH2 group in the positive charge residues, right. If you take the C alpha, right, they may be far, but if you see the charged atoms, these atoms are close to each other. So, in this case if you use any heavy atoms, right, you can get the better information than using either C alpha atoms or C beta atoms, right. Then how to define? So, for example, any atom in a DNA and this protein. So, for mainly in this case, or if in this case you can take any of these two proteins. So, first way is you can just have the complex and cut into two pieces. Now, you got two chains, either one is DNA is here and the protein is here. In this case is the protein 1 and the protein 2, right. Now, you can calculate the distance between any heavy atoms in the protein 1 as well as the protein 2, or in this case DNA and the protein. So, you have the equation to calculate the distance, right, the square root of x 1 minus x 2 the whole square, right, plus y 1 minus y 2 the whole square plus z 1 minus z 2 the whole square, right, calculate distance. Then you keep the distance cut off, say 5 angstrom any heavy atoms or 6 angstrom any C alpha atoms, right, and if this is less than the distance, then we see that two residues are in contact and the two residues may form the interface residues, right, because they share the common interface, one residue from protein 1, another residue from protein 2, because they are very close, right, in space. Fine, how to do that? This is one example for a protein DNA complex, right. So, if you see this is the DNA is here, okay, you can see the DNA here, right, and the protein here, right, and you can see the interface residues, right. You can see the residues marked in green and you can see the contacts which are marked in red, right. So, the green one are the amino acid contacts from the protein side and the red one are the base contacts from the DNA side, right, you can see the contacts, fine. So, now I show the PDB coordinates, right, the PDB which information you will get, right, you get the XYZ coordinates, right, XYZ coordinates, this is chain A and this is chain B, right, okay. Here if you see the complex, this complex interleukin receptor complex, you can say this is only chain A, this chain B for example, right, you have the coordinates here for chain A, this is the coordinate for the chain B, right. Now, if you see the atoms of any residue and calculate the distance with any of the heavy atoms in the second chain and set the distance of for example 5 angstrom, and if this distance is less than 5 angstrom, then you can say the residues in chain A and the residue in chain B are in contact and they are interacting with each other. For example, if you see this 88 origin 88 here, this is the chain A, right, and here this is the aspartic acid 72, right, in chain B. If you get the contacts, right, you can see this should be within the specific cutoff distance. For example, if we take this one NH1 of arginine and the OD1 of aspartic acid, right, what is the distance, right, take this and this. So, 14 and 17 this will be around 9 plus 19, 13 and 10, right, this will be 3, that is plus 9 plus 19 and 21, that is 2, that is around 4, right. So, this will be around 22, this is less than 5, right, fine. So, you can say these residues are in contact with each other, and specifically if you see this is the arginine, this is a positive charge residue with NH1 and NH2, this is negative charge with the OD1 or OD2, right. So, they are having these electrostatic interactions, right. Especially if you see the second one, the NH2 and the OD2 16 and 18 this equal to 2, it is very close, 14 and 12.5 this is also less than 2, that is 4, 20 and 21 that is 1. So, there is very this is less than root 2 plus 2 plus 2, that is equal to root 6, right. In this case it is less than or equal to 3 angstrom, right, it can form the strong ion pairs, right. So, if you see the distance between any two atoms and if it is less than any specific cutoff, right. So, then you can see that this is less than 3 angstrom, okay. You see then this is in contact, right, there also you can see whether they form any specific types of interactions, right. This is an example, fine. So, this is the server for the getting the base amino acid interaction database, right. So, if you give any PDB IDs, you give the PDB code here and you can specify the distance then it asks for the contacts, whether you need the base contacts, right, or the backbone side chain contacts or any contacts, right. You can define from the DNA side which are the contacts you require, right, and the protein side which contacts you like to see, right. Then if you see anything, then you can get the data, right. For example, this is the PDB coordinates for this particular protein DNA complex, right, and here this is the sequence number 1, residue A, right, this is the base pair, this is the second one, this is the protein, okay, this is the DNA, you can see the distance, right. So, in this case you can easily see these are the regions where this DNA can contact with the particular protein, right. This is you can see identify the binding site, fine. So, this is from the distance based contact, right. You can get from this coordinates from the PDB and you can get the distance based contact. So, if you get the information from the PDB, so right, this is the additional information which are included in the PDB, this is called PDB sum. This will give you the pictorial representation of the 3D structures in protein data bank, right. They give you several information regarding one secondary structure and the types of interactions, right, and so on. So, if you go to the PDB sum, you can see the contact between two residues and which type of interactions these residues make, whether electrostatic interaction or van der Waals interactions or hydrophobic interactions and so on, right. For example, if you go to the PDB sum, this is the website for the PDB sum, right. Go to the websites and then you can give the PDB ID, right. If you give any PDB ID here, right, you can give the PDB code, right, and then if you click the submit button, right, then search, then you will get this type of information. This is for 1A2K, right, and you can see this is how they represent the chains B and C, and some of these are the residues, how many residues and how many contacts, right. 70 residues in chain B, 12 residues in chain C, right. They are making about 80 plus 10 contacts, right, 80 non-bonded contacts, right. You can see non-bonded contacts in yellow and the hydrogen bonds in blue and disulfide bonds, this is also yellow, right, this orange, and the salt bridges in red, right. So, you can see here you have only the 80 non-bonded contacts plus 10 hydrogen bonds. Then if you go through the details, you will get the more detailed information, right. You can see the here, this is chain B of 1A2K, this is chain C, right. So, they are making different types of contacts, right. They use different colors. What is the meaning of different colors? For example, this is blue for the this is arginine and like this may be positive charge, and the red for glutamic acid, aspartic acid, this was negative charge, and the grey for hydrophobic, right, and mainly the green for the polar, right. Now, you can see the type of interactions here, right. So, what is this green one, the blue one? The blue one is for the hydrogen bonds, and you can see the other contacts, right, this mainly the hydrophobic interactions, right, because in this case mainly the hydrophobic interactions as well as the hydrogen bonds. So, if you look at the PDB sum, right, so it will identify the probable contacts and based on that classify the contacts in different types of interactions, either electrostatic interactions or hydrogen bonds, right, or non-bonded contacts or salt bridges, right. For any case you can see that, fine. So, now as earlier discussed about the PDB param, it contains more than 50 structure based parameters. We utilized PDB param to get the surrounding hydrophobicity, long range order, right, and different other structure based parameters. We can use the same for identifying the binding sites also, right. For example, if you go to the PDB param and you want to get the binding sites of a particular protein, you go to the website and then just click on identification of binding site, right. So, and then submit, right. Then this will ask you, right, for the distance threshold, which distance threshold you want to calculate the identify the binding sites. So, we see this is 3.5 angstrom, right. So, we will get the interactions like the residue name, here this residue for number 41, this atom name CD1 and chain B, okay, this is from the one chain, right, and this from the another chain, right. So, phenylalanine 72, right, the chain C having a distance of 3.2 kilocalorie 3 angstrom, right, the same 1A2K, right. The tryptophan and phenylalanine, right, they are within the distance of 3.2 angstrom, right, they are having the interactions. The same information you can also get from this PDB sum, they are making the hydrophobic contacts, right, you can get from this information, right, fine. So, in this case you can also get all the contacts, and from these contacts you can also identify which type of interactions they are possibly to make, right, between these two chains, for example chain B as well as chain C. Okay, this is one concept. So, they used the concept of distance, right, distance space criteria to identify the binding sites, right, you can use any distance criteria. So, next concept it is mainly based on solvent accessibility, right. What is solvent accessibility? Right, they are accessible to a solvent. For example, this is a protein, right. So, you can make these residues and atoms in the interlocking spheres. This is a solute molecule. So, you can roll over the residues and atoms, right. I can see how far this can penetrate into the van der Waals surface, right. So, based on that we identify the residues which are in the surface and some residues which are in the core, right. These are the values, right, for each residue. Fine, already we discussed in detail in earlier classes, right. So, how to use the concept of solvent accessibility to identify the binding sites, right. And for example, here show the same figure again. So, this is the complex, which complex this one? Protein DNA complex, right. So, we cut into two pieces, right. Here we are not taking the free protein or the free DNA, take the complex, right, and we made into two pieces. This is the protein part, the same, right, and then this is the DNA part. Now, you can calculate the accessible surface area, right, in the complex state or in the unbound state, right. You cannot say the free state, you can say this is the bound, this you can say the unbound, the same structure, right. So, if you look into these two proteins, right, if you take some region, for example, this region, right, how about the accessible surface area will vary? It no varies, right, because it is also accessible to solvent, here also accessible to solvent, right. In this case you do not see any change, right. If you take this region, right, then you can see this is the complex state, this is the unbound state, which one have a higher value? Unbound, right, because in this case it is accessible to solvent. So, this is a more high accessible surface area than in the complex state, because here it is hindered by this DNA, because this region, right, is covered by the DNA, hence the area will be less, right. So, if you compare the solvent accessibilities with the complex and with unbound state, then if there is a difference in accessibility, then what can we say? Yeah, there is, because of the reduction in the accessibility is mainly because of something else, right, in this case DNA, because of this presence of DNA they reduce accessibility, right, from the unbound state to the from the unbound state to the unbound state. Fine. How to do this? Now, I show an example. Okay, here this is the protein DNA chain. Okay, this is the place where we can see the interacting residues, right. Right. For example, we see this protein. This is the ASA of this complex, right. Isolation 12 has 5 angstrom square and ASP is 55 and so on. Now, this is with the free form, this unbound form, right. So, in the case of proline 45, right, there is defined any difference? No difference. Leucine 46, no difference. But on the other hand, if you see other residues like isolation 12, aspartic acid 13 and 19 valine 20 glycine. So, this is the unbound form. When we make the complex, you can see the difference, right. Then if this is the difference, right, and then we can see that these residues are interacting, right, in the complex. In this case, what are the residues which are interacting with the DNA? Isolation 12, okay, these are the residues which are marked in red. These residues you can find in this region, right, they interact with the DNA. Fine. I show one more example, right. In this case, I put 1 B01, this is the complex, and here this is the free protein, this is the free protein, right. And if you see there are several cases, right, for example, this arginine, they reduce from 255 to 238, right. And some cases it is also possible that the complex case, right, it has more values than the free one. How this is possible? So, here I like to mention the difference between the unbound and the free one, right. In the unbound case we tell you that it is a complex, the complex we cut into 2 pieces and take the protein. In this case if you superimpose this protein as well as the one in the complex, they match, exactly match, right. There is no difference in the conformational change. But in the free protein what we do is we take the amino acid sequence and check for the structures without any ligands, without any DNA. In this case you can see the change in conformation. Because of change in conformation you can see the difference in acceptable surface area, right. Sometimes it is higher in the case of the complex than in the case of this free protein. Then I show an example to the protein interaction complexes, right. So, this is the alpha chymotrypsin and Egg-Linked complex, right. So, this is the protein 1, this is protein 2, right, with the complex and the chain E, right. This is the ASA with the complex and this is the chain E, right. And if you see there are many cases, mainly in this region, interface region, easily you can see the lock and key mechanism, right. If you can see this is the convex and concave surface here, right. So, 12.2 reduced to 0 because it is completely inaccessible, right. Here the 8.15 is completely 0. So, in this case these residues are identified as binding site residues or the interface residues. Also if you see in this chain I continuously a stretch 42 to 48, all these residues, right, they change the axial surface area very drastic change, for 127 to 27, 54 to 4 and so on. So, these residues we identify as binding sites, right. This also you can do in the case of the protein complexes. This is another example, the same I explained earlier. So, this is the complex 1A2K, this is the complex and this is the free protein, right. Some case if you see the free protein higher than this complex and also vice versa you can see in some other cases, right. So, if it is reduced to the complex, right, then you can see these residues are identified as the binding site residues, fine. So, we discussed about 2 different approaches, right. What are 2 different approaches we discussed? Distance based criteria and ASA based criteria. Distance based criteria, how to identify the binding sites? Simply based on the cutoff distance and can identify within that limit. The ASA, how to identify the binding sites? Change in axial surface area, right, between the unbound form as well as the complex form.